Okay. So I'm just gonna have Rabia, Binish, and Faria all speak and tell you a little bit about their mom's cancer journey and what kind of they wanted out of tonight. So whoever would like to go first, you are welcome to.
So much. Thanks again, Allison. Hi, everyone. Thank you again for joining us today. It means so much to us that you took the time to be with us and hear our story about our mother and her journey with cancer. I kind of just wanted to share a little bit about Mom. We're getting started, jumping in with this amazing program with Dr. Santin and Anne's Place. Basically, to put it short, our mom was an incredibly special person. She enjoyed all the small things in life and taught all four of her kids to celebrate the little wins, no matter what they may be. When Mom was diagnosed, that's kind of exactly what we did. We celebrated every bite Mom took of food without getting nauseous. We celebrated the extra lap around the house without getting tired. We celebrated all of her smiles and laughs because we knew in those moments her pain was bearable and she could enjoy herself again. If there's anything that I learned, it was that support and celebrating were going to get us through, no matter how hard it was. Every occasion was an occasion to get the family together, cook a big meal, and enjoy everyone's company. We surrounded and almost at times suffocated Mom with love and affection. A lot of the time I felt helpless because I don't have a medical background. My sisters do, but I come from the interior design and furniture world. So there was a lot that I felt like I couldn't help Mom out with. But I kind of made sure I fit in some way as one of Mom's main caretakers. But I quickly learned Mom just wanted to hang out with her kids and be around us more than anything else. So that's what we did. We spent every waking second with her. When she was up, we were up, even if it was at 2 a.m. and she wanted a snack. We would sit with her at the dining room table, their heads in our hands, just listening to Mom enjoy like a cracker or two, whatever she could really stomach at the time. If I could give any non-medical advice to someone struggling with cancer, it would just simply be to be present for the ones who are fighting. And if you are the one who's fighting, finding a community like Anne's Place that's present for you. I think our biggest regret is we weren't able to bring Mom to Anne's Place sooner, but we're thankful we were able to create a circle of people around her and lift her up. Finding her the correct care team and medical team, that was our biggest win. And again, we just want to thank everyone in Anne's Place, Dr. Santin, for the opportunity to be here, talk about Mom, and just spread her special story.
Hi, everyone. My name is Rabia. I am Faria and Binish's older sister. My sisters are extremely humble and not talking about all the details and all the things that they did, stopping life and dropping everything to be there for Mom. I wanted to give you guys a little background about Mom and what kind of diagnosis she was and what our journey was like. I'm the healthcare professional in the family along with my other sister, Binish. I'm an occupational therapist, and being an occupational therapist and being in healthcare treating cancer patients has been part of my career for the past 20 years. Never did I think that I would be taking care of my mom, who would be going through not only cancer, but one of the most rare and aggressive forms of cancer. So a little background about our mom. My mom was 58 years old, living a normal, healthy life. The pandemic struck. It was April 2020. We'll never forget the day. It was just when the world was shutting down, and our mom woke up in one morning and said that she was having some pain in her stomach. If anybody knew our mom, you knew that she would never complain about anything. Everything to her was putting her children, her husband, her family first. And never did she ever complain in my entire, I'm 39 years old, life that I've ever heard her complain about anything. So for her to complain and say that she was having stomach pain and that she was going to the doctor was pretty alarming. But then again, we were just thinking it could be anything, you know, and her symptom that she at that time expressed to us was that she was having bloating for 2 weeks, and she felt bloated and gassy, and she was just taking some over-the-counter gas medication, not thinking anything of it. And of course, she didn't tell us any of this. She had this for 2 weeks, just the bloating, and then all of a sudden woke up with the pain. Long story short, she went to her primary care doctor. Her primary care doctor was a little concerned and said, I'm going to send you to the emergency room. It was a Friday afternoon. I'm going to send you there to get a CAT scan. I'm not really sure what's going on with your stomach and why you're having this pain. So she went to the emergency room where she had a CAT scan done. Being the height of the pandemic, everybody was in the parking lot waiting to hear what was going on because we couldn't be inside the ER with her. So we waited and waited, and we got the worst phone call of our lives, that our mom had cancer. And not only did she have cancer, but they found it spread all throughout her pelvic and abdominal cavity. And they weren't really sure how severe it was. They knew it was severe, but didn't really know. At that moment, they just wanted to stabilize her. Her stomach was bloated. She was having a lot of pain. Her blood pressure was high. Her body was not normal. Something was off. And it felt like a switch had gone off. Like we went from hiking a week ago with mom and enjoying the outdoor weather because there was a pandemic and there was nothing else we could do, to all of a sudden, mom is in the emergency room and she has cancer. So from that moment on, we knew that, as a healthcare professional, I knew that we just had to take action. There wasn't time to wait. There wasn't a moment to waste. So we got her to our local oncologist right after she stayed at the hospital over the weekend. She got home. We went to the oncologist that Monday morning, and they said she's got stage 4 or 5 cancer, but we don't know what type it is. So they did the process of all the genetic testing and all the things they had to do. And they said, what we have to do right away is chemotherapy. We have to start right away. This has spread and we need to get a handle on this. So we knew this was a very much life-threatening situation. And at this point, our whole world just stopped. Everything stopped. The world was already stopping with COVID. But on top of that, we just knew that this was a major, major situation. So mom started off with chemo, and chemo, she went through a lot of different chemo. We started getting the results back from her testing, and we found out that this was a carcinosarcoma. So the origin being uterine or ovarian. So, but it was one of these really rare and aggressive cancers. The good news we finally got after months of chemotherapy, and chemotherapy was a very hard time for the whole family. Obviously, to watch your loved one go through it was absolute torture. During this time, mom went through all the ups and downs of the side effects of chemo on top of everything that was going on with the tumors that had spread. She was having paracentesis weekly at the hospital. And during this entire time, she wasn't complaining. She was worried about us. She was worried about us dropping everything to help her. And that's who she was. She was selfless the entire time throughout this journey. As all this was going on, we knew that there had to be more out there. We had to figure out what we could do to help her. And the process started where all of us divided up between us 4 siblings, my sisters and my brother. We also spoke to a gentleman from Australia who specialized in the Gerson protocol. So we spent 3 hours talking to him about what we could do in terms of natural remedies and homeopathic remedies. With our brother being a nutritional therapist and personal trainer, he started implementing some of the diet changes and exercise to help. So the chemo would also help, intermittent fasting. And between reading up on all these things and then actually doing my own personal research, as I was taking care of my mom during this time, I was treating 2 other patients of mine who had gynecological cancers. And I was comparing notes to how their treatment was going and how they were reacting to what they were going through, and using that as a guideline and a basis to compare what mom was doing. And during this time, we started doing more research. We found, looked for support groups, and we found a wonderful support group on Facebook with women who were going through had the exact same diagnosis as our mom. And that support group became one of our biggest support systems for our family, cheerleaders for our mom. On that support group, we would ask them what they were going through, how they were managing certain symptoms. And between all the information that we were getting from research, between what my patients were doing in healthcare to studies that were out there to this Facebook group, we were getting a lot of emotional and actual support, medicinal support. And we opened up another avenue. We looked into the avenue of CBD. And being, you know, mom was a traditional Pakistani woman, it was a very hard question to ask her, would you consider medical marijuana? And it was something we didn't even want to ask her. We were almost embarrassed to ask her because we didn't want her to think we're offering her drugs. But we did our research. We found out, and we, even with patients that I had who were taking CBD, it was helping with their nausea, it was helping with their pain, it was helping with their sleep, it was helping with their anxiety, and all the things that come with the diagnosis of cancer. And we were able to get her a medical marijuana card, and that was a whole process. But we learned so much throughout this process of all the different avenues and all the different pieces that may help that one person. But it took a lot of work, and it took a team effort. It was a team effort between all our family, and us even reaching out to extended family members and doctors and oncologists that we knew, friends of friends in other states, and what they were doing and what they would recommend. And our research never stopped, and we never stopped looking for finding the right path for mom. During this entire time, as mom started doing chemotherapy, we started getting some good news. So she did chemo for a few months, and as her diagnosis was in April, we realized that the chemo was working, and her tumors were sensitive to the chemo. So her tumors were shrinking, which was wonderful news. And like my sister Fadia had said, every little milestone, every positive news that we got has to be celebrated. It has to be, whether you're going through it or you're supporting someone going through it. That was the only way to survive. And it really helped with mom's mental health, making her positive, keeping her positive, letting her know that, you know, all this fight that she's doing, that we're gonna find some sort of positive solution out of all of this. So after she had all her chemo, they went in for surgery. So she had her first surgery September, October of 2020, where the surgeon was able to remove all the tumors. So they actually got everything out, which was amazing. So the only thing that was left that they couldn't get to was a region near the liver, a tumor that was left there, and some parts of a tumor in the pelvic region. But overall, they got everything. Her tumor markers came down. She was feeling great. She recovered from the surgery. And it was something that we were all just so grateful for. After that, they continued with maintenance chemotherapy. So she did that for a few months, and things were looking pretty good. But of course, she was still very weak, and she was deconditioned. Her body had gone through so much. But once again, she got up, and she was there for every family birthday party, every occasion. She wanted to be present, and we wanted to make sure that we were savoring all those moments. As time went on, a few months after surgery, we realized they did another scan that there was another tumor growing in the pelvic region. So this news hit everybody hard, and obviously it was very upsetting for mom because we thought we were heading in the direction of being clear. So she started a different chemotherapy. And it was a couple other regimens, which, you know, I don't know off the top of my head, but she started doing more chemotherapy. And at that time, the tumor was not really shrinking. So they decided to go in for a second surgery. So this was almost a year after the first surgery. So she had her second surgery in 2021. And that was about October 2021. And at this time, they got the tumor out and everything looked good. And this was the closest we were to hearing the word maybe remission. That was something we're all praying for. But at the same time, we knew the reality of this cancer. We knew the reality of this disease, and we had to make sure that we were not letting our guard down. So we were still making sure that mom was doing all she needed to do in terms of eating the right things, taking care of herself, being there for her emotional, mental health. We did not want her to lose hope, and we did not want her to get down. And that was a big part of all of our jobs, not just managing her medical symptoms, but making sure that emotionally she felt the love and support. Because as we know, a lot of our health and a lot of our physical is tied into our emotional. And if we can provide that emotional, moral support, it really helps the immune system, and it really helps somebody feel like they can do it. So at that time, mom had her second surgery, and they started her again on maintenance chemotherapy just to get rid of any other cancer cells that might be floating around. And at that appointment, mom had asked the doctor, like, hey, you know, maybe I can get my port out. Like, you've got everything else out. The surgery went well. And the doctor was very pleased with everything. And this was, October 2021. About 2 months later, end of December, beginning of January of this year, they did a further scan and they found another tumor in the pelvis. And this was very disheartening because at this point, we were just very much told that this cancer was rare. This was aggressive. They've tried all the chemotherapies and nothing else was working at this time. But we didn't want to stop and we were not going to stop. And mom didn't want to stop. So we kept doing what we needed to do, whatever we needed to do. So at this time, we started branching out. We went and got second, third, fourth opinions. We went to Memorial Sloan Kettering. We went to Hartford Hospital. We went to St. Francis. And then we came across Dr. Santin. Dr. Santin, we found out through the Facebook support group, through a website called the GCS Project. And now Allison will post all of this information that we're sharing for the support groups and the websites at the end of the presentation. We found them. And at that point, it was a very hard time in mom's medical treatment. Her tumor was growing 1 centimeter a week. And it was really getting to the point where it was growing extremely fast and things were getting really out of control. And we really felt like we didn't know what else to do. But we were there with mom every second of the fight. Excuse me, sorry. And in April, we reached out to Dr. Santin end of April. And at that point, mom had to go into the hospital because she developed a bowel perforation. So what that exactly is when the tumor itself is now expanding and sticking to parts of the colon and parts of the intestines and causing holes, which then resulted in sepsis. At that point, we had to rush her once again to the emergency room. And this time we went to Yale emergency room. And we were under the care of Dr. Santin's team and the wonderful people at Yale. And they stabilized my mom. She was septic and very sick, very, very sick. And it happened overnight. And they stabilized her and she went into the ICU. She came out of the ICU and she was still smiling, not complaining. And we were there every second with her. And Dr. Santin and his team were able to do some further testing. And they did some more testing with mom's tumors and some of the genetics. And, we came to the results that at that point in time, there was nothing available. There was no clinical trial available. There was no other treatment available. We had kind of exhausted everything that we could. But at the end of the day, I know my sisters and I, my brother, my dad, we knew that we had left not one leaf unturned. We did everything that we could, and we gave mom everything that we could with all our being. And for that, we are grateful, and we're grateful to Anne's Place. It's to be able to offer such a thanks for the community and for people that are out there. If there's anything that we could do to help you, because we have learned a lot during this process, and our goal is to help just one more person. And I apologize, but thank you. I'm going to give this to my sister Beanish now.
Hi everyone, my name is Beanish. I am a licensed social worker. So similar to my sister, I think a lot of the work that I did with my mom was just supporting her emotionally, as best as I could, not as a patient, but just as my mother. Unfortunately, during COVID, we were not able to visit Anne's Place, but we did find ourselves there a week after her passing because we knew that what she went through was remarkable and we wanted to share her story as well as our journey alongside her. So I think my sisters did a really good job giving you a recap of what, the 2 years were like for us and being able to be her caretakers. we honestly wanted to share her story because there are so many other women, mothers, grandmothers, daughters, friends who have suffered from this cancer, and they may not have the same resources that we found along like our journey. And so we really came to Anne's Place to really be able to provide those resources and that support to other families who may have gone through the same thing that we went through. I think, like my sisters noted, my mother was a selfless human being. We were very lucky to have her for the time that we did. And as her daughters, we want to continue her legacy. So we want to be able to give back to people. We want to be able to provide support for people. And we're very lucky to have come across Anne's Place and Dr. Santin to be able to do that for, even one person. I think, what we've learned is this cancer is very aggressive. It's very scary. There's a lot of unknowns and a lot of uncontrollables. But I think similar to what my sister Fatia said, we celebrated all of the small wins with her. And with our family, and she celebrated them with us. So she tried to be as present as she could for those days and those moments. And, we've learned that although we did not know what we were doing throughout the process, none of us really had any idea. We somehow figured it out along the way. And like I said, I think we're very grateful to have a space like Anne's Place to be able to give to families who maybe don't have the same support that we have for one another. We're hoping that today's presentation, with Dr. Santin, who we're very grateful for, we'll be able to provide some information and shed some light on, what this cancer is, what it can do, but also what are the steps to be able to, work through and find remission. We're hoping that, our mom was unable to, and tomorrow is her 6-month anniversary since her passing, but we want people to be able to get to that place that my mom couldn't. So, thank you for having us. Thank you for giving us this platform, and we're looking forward to Dr. Santin's presentation.
Thank you so much for sharing that story, being able to share, a painful yet beautiful story, because there's obviously so much love. And thank you so much for making this go into motion so that people can get informed and hopefully have some clarity. So I just need to do a few little tech things here before I hand it over to Dr. Santin. And I'm going to move spotlight. And I'm going to make Dr. Santin a co-host. And we are still recording. So Dr. Santin, Dr. Alessandro Santin, he's professor of gynecological oncology, Department of Obstetrics, Gynecology and Reproductive Services at Yale at Yale University School of Medicine. We're feeling very, very blessed to have him here and for him to share his expertise. So Dr. Santin, I will hand it over to you. I'm just going to spotlight you. And you are all set.
Perfect. Thank you. So let me open the presentation that I have ready here for a second. So can you, let me see. Trying to share. Do you see my screen?
We do.
Okay, perfect. So let me put it in slide presentation, and let me reduce this. So, okay. So first of all, thank you for the invitation. Thank you for having me. As you heard, I was somehow present only in the last really a few weeks of this journey. And that, of course, is unfortunate because as a physician, as you know, I have the so-called bodyguard syndrome. I really always try to do my best to save my patient. And unfortunately, here I was involved after this sudden bowel perforation that really prevent me from helping her in the way that I wanted. But I was anyway able to get in touch with this fantastic family, and you heard the 3 daughters here, and that is why when they asked me to do this, I was really happy to talk to them, because of course this is for them and to all of you about the type of work that we're doing at Yale to really try to help a patient that they get diagnosed with this this rare but extremely aggressive gynecological cancer. So without further ado, let me tell you a little bit about what we're doing at Yale. So let me start with the minimal, background here to give you a little bit some idea of what we're dealing with. So as you an oncologist, I know that over 100,000 women in the US alone are going to be diagnosed with a gynecological cancer this year. And unfortunately, even when these women are treated with the gold standard therapy for gynecological tumors, namely surgery, as you heard, chemotherapy or radiation, over 1/3 of these women will die from this disease, regardless of what we're trying to do. So, so this is telling you that of course, there is still an unmet medical need in dealing with this rare tumor, this aggressive tumor. But again, let's go back to the background here. So this tumor here, when this patient was referred to me, was initially defined as a potential ovarian carcinoma. But after my interaction, my review of the pathology, I realized that most likely this was really uterine cancer metastatic to the ovary. And that was something that, of course, to me, and I did not really share too much of the information, changed the picture, because, of course, uterine cancer, at least at Yale, we had trial for uterine cancer, carcinosarcoma, but at that time point, there was no time to do anything, as you heard, because the situation deteriorated very quickly. Now, uterine cancer is the most common gynecological cancer. Over 66,000 American women are going to develop uterine cancer this year, and the annual death toll is close to 13,000. Carcinosarcoma account for a small minority, so about 3 to 5% of all this uterine cancer. But because of the biologic aggressiveness, about 20% of all death related to uterine cancer are secondary to this rare tumor, carcinosarcoma. Why are they called carcinosarcoma? It's because they are biphasic tumor, means that they have 2 different types of cell that they make this tumor. The epithelial component that are the most common, right? The epithelial cancer are the most common cancer in the uterus. But here there is also a secondary component that is a mesenchymal sarcomatous component. And that makes the situation more complicated, because now you're dealing with a cancer that has really 2 different types of cell inside. And this is also a problem because, once again, when you're dealing with this tumor and you're trying to look for new drug or new trial to offer to this patient, because they are rare, because they have these 2 components, patients with carcinosarcoma are rarely eligible for clinical trial. And that is why you really need investigator, clinical scientists that they have a specific interest in this malignancy to design this trial, because otherwise, once again, large pharma-sponsored trials are usually not available for this patient. Other few things to make it a little bit educational for you is that these tumors that are rare are anyway more common in African-American women when compared to Caucasian. And we don't really know why, and we're trying to understand, and we look to the genetics of this tumor to understand why. But this is a reality, 3 to 5, 1 to ratio. How are we dealing with this patient? In the way that they just told you. We immediately try to do the surgery. Sometime, like in this situation, the disease was already spread out outside the uterine cavity. And that is why we in that case, we recommend neoadjuvant chemotherapy. So if you were chemotherapy before doing the surgery, with the ultimate goal to shrink the disease to the point that you can really do the staging, hysterectomy, and laparotomy. And after the surgery, of course, either the chemo or some more chemotherapy, like in this case. But the problem is, as you heard, and this unfortunately is not the exception, quite often these tumors, they are rapidly become resistant to chemotherapy. And therefore, we really are, there is, again, a desperate need to develop new treatment for this patient. Now, one of the questions that a lot of my patients ask me is, why did I develop this cancer? What happened? And the answer for carcinosarcoma is not completely clear, because there is a subset of these tumors I'm going to show you in few slides that that really are developed in women that are predisposed to develop uterine cancer because they have inherited a genetic alteration from either their father or mother. But the majority of carcinosarcoma are called sporadic, meaning that there is not a genetic link to father and mother, so there is no familiarity. But the tumor now arises because of the transformation of the cell that acquire genetic mutation during the lifetime of the patient. And here, again, briefly, I listed the different type of alteration that I see in my patients. Sometimes I see mutation in oncogenes, where a single alteration can really affect the cell to the point that the cells start dividing very quickly. And this is, again, when with my student, I do the analogy with the car. This is like when you have an oncogene that is mutated. It's like when you have a stack accelerator in a car. The cell, again, like the car, keep on moving very, very fast, and you cannot stop that cell, keep on dividing. Sometimes the tumor instead is acquired because of a mutation in other genes that are called tumor suppressor genes. These are really the genes that can be inherited. And usually, again, we inherited one, either from the mother or the father, but the other one is working. So that is why in this case, women, they have a normal life. But when the second, again, gene that is the healthy one, during the lifetime acquire a mutation, now both genes, they don't work anymore. And that is like a dysfunctional brake in a car. Now we cannot stop the car. And in this case, the tumor. And finally, the third type of gene that can get altered and cause cancer, including carcinosarcoma, are what we call the stability gene or caretaker genes. And these are genes that usually repair our DNA. In our everyday life, our cells get damaged. And this may happen in multiple ways. Our diet, we don't know what we're eating, right? What there is inside that can cause mutation, alteration, or we are exposed to different type of radiation, different chemical. They affect our cell. They may cause DNA alteration, but we have genes inside our cell that are usually able to repair this damage that happens on a daily basis. But sometimes this mutation take place in this gene that repair DNA. And these are, again, the caretaker genes in our cell. And when that happens, it's like when you have really an inept car mechanic, we cannot fix the car. And here we cannot fix the cell. And the cell, again, keep on replicating and becomes more and more aggressive. As I told you before, some of my patients, they develop this type of cancer because they have inherited an alteration from their father and mother. And here I wanted to show you again the picture of Angelina Jolie, this famous actress, because again, she got public a few years ago. She knew that her grandmother and mother, they died for both breast and ovarian cancer. She tested herself. She found herself being the carrier of the BRCA1 gene alteration, and she immediately acted. She said, I don't want to die like my mother or my grandmother. So she had a prophylactic mastectomy, and after that, she did again a prophylactic oophorectomy, a salpingectomy, with the ultimate goal to decrease the risk to develop, again, ovarian cancer. Now, with this type of background, let me tell you a little bit more about what we are today able to do. So now that we know that the cancer is a genetic disease, and we know how to, to sequence this tumor, as I'm going to show you in a minute, we now have a different way to deal with this patient. What does it mean that? It means that when I know that my patient has specific alteration, genetic alteration, now I can really understand if this tumor could be susceptible to a specific targeted agent. And if you were, the one-size-fits-all approach of chemotherapy that is currently still the gold standard, as you have heard, again, it may help and help several of my patients, but in a biologically aggressive cancer, quite often the tumor come back and become resistant to chemotherapy. And that is really when we need to look in another direction. We need to really look, in my case, I'm trying to explain to my patient outside the box of the chemotherapy. We need to look to personalized treatment modality. And that is really what we're trying to do at Yale in my division. And I'm going to show you what does it mean to treat patient using this customized type of treatment. So what I'm going to show you here is what I believe is the future of oncology, not only for patients that develop uterine cancer, carcinosarcoma, but really any cancer that is biologically aggressive. So as a surgeon, the first act is usually surgery. We want to remove all the disease that is, again, visible, present in the abdomen of our patient. And that is why we try to be aggressive from a surgical point of view, try to remove, again, the primary site of the disease and any metastatic site. And at that time point, we send, right, the tumor to our pathologist. The pathologist, after 7, 10 days, 2 weeks, come back with a report and is going to tell us, this is a tumor that started in the uterus, is a carcinosarcoma, has a high-grade serous component, has a mesenchymal component. But again, this is something that 30 years ago I was already getting, and it doesn't really help me much to treat the patient, with the exception that now I know what I'm dealing with in terms of, again, site where the disease, took place in first place, but really not much more than that. That is why we need to do better. And what we do at Yale is the following. A piece of this tumor, when I do the surgery, patient sign a consent before I explain everything to my patient, when I have, again, the possibility to do it up front, right? And a piece of this tumor goes to my laboratory at Yale. And at that time point, my postdoc, they extract the DNA, they extract the RNA of this tumor, and they send it to the West Campus facility, where we sequence the entire genome of the tumor. So we sequence over 21,000 genes, all the genes that encode for a protein. So for a function of the tumor, we sequence it. And the ultimate goal here is to understand the genetic landscape of the disease, is to understand what are the driver mutations that have caused this cancer, with the ultimate goal to take advantage of the knowledge to really use as the Achilles heel of the cancer. So to go after those mutations with specific new drugs. At the same time, a little piece of that tumor is also placed in a petri dish to try to establish a primary cell line so that the tumor can grow in the lab, in the incubator. And another little piece of the tumor is established as a xenograft in a SCID mouse. So a mouse that has no immune system and has been really, this mice have been bred in this way to allow, again, in this case, a tumor from, again, a human being to be established in their body. These mice, these are called patient-derived xenograft. And these are also called avatar animal, right? Because this is the piece of the tumor of the patient that is now growing in a complex model, that is, in this case, is a SCID mouse. And growing in the animal may allow me to study much more, right? Because when I have the sequencing of the tumor, and now I have identified potential target, I can immediately try to understand if the specific drug with specific antibody is effective in that tumor for that specific patient of mine without doing that to the patient right now, because I'm not yet sure if it's going to work or not, right? So I'm using this model to increase the chances of success in the patient, because I know that if I'm able to stop the tumor in the SCID mouse when she's getting the gold standard treatment, that is the chemotherapy, right, to be ready for what is going to happen after, unfortunately, the majority of my patients, I know that if I'm able to stop it with the targeted treatment, I can offer this to my patient. And I know that if there is any toxicity, it's really worth it, because I know that 90% of the time that I see a shrinkage in the mouse, this is going to be also something that I'm going to see in my patient. And the last thing that we're doing at Yale, that, this is not yet standard of care worldwide, but we have the possibility to do it in our institution. Now that we know the genetic landscape of the disease, so we know the mutations that are present in this cancer, we can also follow the progression in my patient using not invasive method, such as a collection of few cc of blood every 3 or 4 months. Why am I be able to do that? As you know, one of the most difficult questions to answer to my patient or to our patient in general is after the surgery is, a lot of my patient, the family, they're asking me, Dr. Santin, have you been able to take everything out? Or during the chemotherapy, when I do the CAT scan and I see again that there is no more visible disease, the family and the patient is asking, are you really sure there is nothing left here? And again, we do the best that we can. We use CAT scan, we use MRI, we use PET-CT, that are the most sophisticated and sensitive methodology that we have currently available in terms of imaging to try again to find if there is anything left, right, in terms of tumor cell. But this personalized diagnostic modality that I just told you, the possibility to go after the single mutation present in the bloodstream of the patient is thousands of times more sensitive than the standard imaging, because I'm able to detect one single fragment of mutated DNA, and I know which one it is because I've done the sequence of the patient, so I know what are the potential mutations that are circulating in the blood of my patient. And using this raindrop digital PCR that is available in my own laboratory, I can identify one piece of this mutated DNA among 1 million of a normal DNA present in the blood of my patient. So this, of course, this is the future, but is already the future that is available right now at Yale and in some other institution in the world. Now, one important thing that I have to emphasize is that when I do the genetic sequencing and when I find again mutation that may help me to personalize treatment to my patient, I cannot take for granted, even when there are drugs that specifically target the mutation, that is going to work in that specific patient. So that is why, once again, we need this model. We need this avatar animal in particular. And how does it work? One more time, I do the surgery, I do the resection. Part of the tumor is placed again in the avatar animal. During this time period, my patient now go through the standard of care, right? That in many of my patients is after the surgery, chemotherapy, followed by radiation. But during this time period, I'm sequencing the tumor. I'm getting information about the genetic sequencing and the potential mutation. And at this time point, I'm able to see if there are druggable alterations present. And there are more and more every week, every month, every year, we find more and more druggable mutation available. At this time point, now I have the time to treat the animal here, right? As the tumor of my patient, I found a mutation that could 1 or 2 or 3 can be druggable. And I try to treat the tumor of the patient in the avatar animal. And if I'm successful, now I can go back to my patient and say, you know what? We have treated this number of animals. We have seen that the tumor is sensitive. You have a recurrent tumor, is now resistant to chemotherapy. This is what we can do for you. And again, this is something that we have been able to accomplish because thanks to, again, grant that I've been able to obtain through the NIH and the philanthropic gift of some of my patients, some also that are watching this presentation, I was able to sequence for the first time hundreds of carcinosarcoma and uterine serous carcinoma that are the precursor of the carcinosarcoma. And that allow me, right, not only in a single patient, but in hundreds of my patients, to identify the pathway that I show you here of the most common alteration present in this aggressive tumor. And some of these alterations, of course, such as the RB2 that I'm going to tell you in a minute, but there are others, of course, I don't have the time to tell you everything tonight, that could be really druggable for my patient. Let me tell you about the HER2 story because it's a happy end story, but there are others, I told you, that are evolving as we speak. When I found that RBB2 was highly amplified, so altered in a large number of my patients with carcinosarcoma and uterine serous carcinoma, I immediately thought, can we target this, oncogene, this mutated alteration? How can we do that? There is an antibody called trastuzumab over Septin that has been, again, developed many years ago in breast cancer and was very active in the subset of breast cancer patients. They have the same alteration that I found in my carcinosarcoma, uterine serous carcinoma patient. So my thought was, can we try to use trastuzumab in my patient when, again, either they failed chemo or in combination to chemo to make the regimen more effective? So the first thing that I did is exactly what I already told you 2 or 3 times. I took the tumor of some of my patients that I did surgery at Yale, or some of my colleagues that, of course, rely on me for the translational work that we do at Yale. I produced the avatar animal. I tested the animal. And let me give you an example here. So this is the avatar animal. This is with a large mass, as you can see here. This is, again, was a carcinosarcoma patient of one of my patients. And I treated, again, multiple animals with 3 different things. I used the control that was the placebo, right? The nothing else than saline to understand if this tumor was able to grow. And as you can see, all animals in about 60 days were dead because of the expansion of this tumor. And after that, I treat 2 other things. I treat trastuzumab and TDM1 that are this antibody targeting HER2. And as you can see here, I saw this amazing response. I was able to make disappear the tumor in 3 weeks. So I said, you know what? It seems to really be working well. But 1, 2, 3 patients are not enough, right? In science, we have to do clinical trial. And that is what I have designed. This was an investigator-initiated study, supported in part by Genentech and in part by, again, the philanthropic support of some of my patients. And I did the proper thing. I divided my patient into groups. Half, they received the gold standard. There was the chemotherapy, the carboplatin and paclitaxel that is considered the first regimen to use in the treatment of this patient. And in the second group, I was given the same chemo plus the trastuzumab, this antibody, with the ultimate goal to see if I could, increase the overall survival of my patient, as well as increase the progression-free survival, that is the time disease-free that my patient may have after the surgery and after the completion of chemotherapy. And very quickly, let me show you here what we've been able to accomplish. Adding trastuzumab to the chemotherapy, I was able to double the progression-free survival of my patient. This was unprecedented because in this aggressive tumor, usually an increase of 1 or 2 months is already a great success. Only adding an antibody that was highly targeted against this amplified oncogene allowed me to double progression-free survival and increase the overall survival of my patients significantly when compared, again, to the group receiving chemotherapy alone. And now, the important thing about this is that because of the success of this study that was published on 2 of the most highly respected journals in the world, the Journal of Clinical Oncology and Clinical Cancer Research, now this regimen that we developed at Yale, doing the thing that I told you, is now the standard of care worldwide and is, again, the regimen that is recommended by the National Comprehensive Cancer Network. That are, once again, the guidelines that are recognized as standard of care in oncology worldwide. And now thanks to this, we are able to offer this treatment and this pay and reimburse by insurance company in the US and in majority of the rest of the world. So this is an example, as I told you. I have 3 other examples like this that I've been able to show you using other antibody targeted different antigen, such as the TROP2 antigen that is currently in phase 2 and phase 3 clinical trial at Yale as investigator-initiated trial and is already positive. So I'm going to move to the second stage because I've seen amazing responses, my carcinosarcoma patient and my uterine serous carcinoma patient, as well as another antibody drug conjugate that is targeting folic acid receptor alpha. That is another receptor that is highly expressed in carcinosarcoma. So these are new treatments that are already in the clinic that I'm able to offer to my patient, as long as, of course, they express this type of alteration. But I'm looking in all my patients to see if this alteration are present so that I can offer this additional hope of treatment and, again, prolongation of life and possibly cure to my patient. And I'm going to stop here and then wait for your question if you have any.